Welcome, everyone. Thanks for being here. Today, I am very glad to. Oh, is there something else first? Okay. I am very glad to introduce Professor Russ Corbett-Detig from the University of California, Santa Cruz. And I have known Russ for a little while now. Ever since he was a post-bac following his undergrad at UC Davis, and I was a postdoc in the same lab. And from there, Russ took off to begin his PhD work at Harvard in the lab of Dan Hartle, on both a bachelor and PhD alumnus of UW-Madison in this department. In the Crow lab. He thrived in that environment, and then did a postdoc with Rasmus Nielsen at UC Berkeley, which I'd also done at one point. Then at I don't know exactly how young an age Russ began his faculty position, but definitely younger than I started here. I've continued collaborating with him, through probably each stage of his career, which, says something about just, how useful and valuable a collaborator Russ is. And, he's helped me with everything from, thorny computational analyses to, fetching some more flies for me from places like the Ethiopian highlands. It's not just, a broad array of skills that he has. He has also demonstrated the ability to make substantial scientific progress in kind of a striking range of topics as an independent group leader. So we'll get a taste of that during his visit here as he gives two talks, one here and now and one through the evolution seminar tomorrow at 12:30 in this same room. So tomorrow's talk is about COVID phylogenetics, tracking evolution through the pandemic. And today's talk, well, you're about to hear about it. It's about transposons and the origin of introns and what's going on with those elements across the tree of life. So please join me in welcoming Russ to Madison.
Yeah. All right. Well, thank you, John. And of course, thank you all for coming out. You may have noticed I've switched the title slightly, and that's because I've done a bait and switch with the last third of this because I submitted a aggressive abstract that I couldn't quite deliver on. So there's a slight difference at the end here. I hope you'll enjoy that as well. And we'll get going. I'll explain a little more as we go. There we go. Okay, so I have been starting talks with this slide, and it's still true for some time now, that evolutionary genetics has a really long history. I mean, John just referenced Jim Crow and a bunch of other people that worked here ever ago and till recent times. And it's had lots of different people thinking about these kinds of problems for quite some time. And just two of them that are sort of the most relevant to the talk today would be Dobzhansky and Muller, who started thinking about genome structure very, very early in the process. And as with a lot of new fields, you had lots and lots of questions. I think evolution was a field dominated by theory for a very long time. And so you had lots of the foundational, like central questions laid out all the way back in the 1920s. So what is the role of genome structure in evolution? Is genome structure itself selective or functional? Is it important in some way? But as with a lot of relatively early fields, you didn't have the data, right? And that's been the big revolution of the past decade or two now, where we have genomic data now at an incredible way to start going back and asking these questions with increased precision and detailed insight into real natural processes. And that's kind of the big thing that's driven this part of my research program for quite a while now. And of course, just to emphasize that this has been ongoing. It's not just stopped a couple of years ago when genomics got to be out there, but even recently we've had major advances. So the first telomere genome assemblies really ushers in an even new era of cataloging complete genomic variation, and the ability to actually accurately measure these things with this kind of detail. And so what that means, though, if we can now measure these things, is that one of the big questions remains. It's the real problem is, can we interpret genome structure? And I think this remains probably one of the major frontiers of biological understanding, is how do we actually go in and look at genome structure and understand what it means to us when we do measure these things in the case of telomere assemblies or just species across the tree of life. Okay, and so with that, I'll tell you about two things. I mentioned a bait and switch here. So the first part of this will be all about work on the origin of introns, kind of try to answer the question, where do introns come from and how do we know? And what are the mechanisms that they arise in new genomes? And the second is going to be some older work that John and I were just talking about, the gene expression and chromosomal inversion. So how can we learn experimentally about what to expect when we have genome structure changes, what should happen to functional aspects of the genome sequence? Okay, so starting with the first one, introns are an old mystery, like all these things. And in fact, when they were first discovered, it was actually something no one had really predicted in the early molecular evolutionary biology days, because why do I need to do this? Why does a gene need to be broken up into little bits? And that spawned a whole series of articles with almost the exact same title of Why Genes in Pieces Revisited. And I've tried to do the version of this slide that has like 20 of these, but it gets a little overwhelming. But take my word for it, we had the exact same title over and over and over again because we didn't have the answer, right? We had lots and lots of theory about this, but actually, I think relatively few clear understandings of where these introns were coming from, why they might be in genomes. And of course, since then, we've come up with quite a lot of ways that introns are important. So just two of the nice examples here. There's actually a ton of others. So you get alternate splicing of the double sex gene in Drosophila, which basically determines whether an individual develops into male or female, with a bunch of other stuff. And enhancement of expression. So lots of introns massively affect gene expression levels because they recruit promoter sequences or the cis-regulatory elements in them. But of course, those are functions that would have to arise after the intron exists. It's very unlikely it inserts and immediately is good at determining the sex of the individual. And so that's selection on the variation after it exists. Where do they come from in the first place is the big question. And so this is probably the prevailing theory on the topic. It's work from Mike Lynch, and of course it's Mike Lynch, so the explanation is the effective population size, where the idea here is that he's showing you a proxy along the top of the effective population size. That's essentially the amount of genetic variation within a population. And he's showing you that the mean intron size increases as you go up as you go down in population size. It's oriented the other way. And the number of introns per gene also increases as you go down in effective population size. And so the idea here is that introns are weakly deleterious. When they arise, they go to fixation in a smaller lineage, more likely than one of the larger population size, because natural selection would purge it from that larger population. This, of course, was met with skepticism pretty early on in the process, and it actually it's tricky to explain much of the overall signal with this sort of thing, and there's sort of a couple ways you can look at that. One is to do ancestral state reconstruction. When you do this, you see that the last eukaryotic common ancestor almost certainly had tons of introns. That is to say, we have introns at the exact same positions in bunches of genes across eukaryotes. Seems like that probably came from the common ancestor. And then maybe a little more directly, if we look at populations that we think have low effective population sizes, things like mammalian species, comparing in the early days mouse and rat, human and mouse, we see there are absolutely no gains you can find when you do that kind of comparison. There were a few losses, but it's extremely, extremely hard to find gains. And human and mouse are actually, they're pretty distant. They're 60 million years, something like that, divergent from one another. And so it's a little bit surprising if that's not happening here. But of course, if we zoom out even further than that, and we just kind of look across eukaryotic life, on the one side, we see things like Saccharomyces cerevisiae here, where there's basically no introns or very, very few introns. On the far side, things like humans, where there are many, many introns per gene. And so this isn't the fully phylogenetic context, but you can kind of extrapolate from this that there must be sort of dramatic gains and losses occurring across eukaryotes, where you couldn't get this much dispersion in the number of introns in genomes, right? And so that tells us there's something going on that there's a process going on here that maybe we don't fully understand. And I'll freely admit up front, there are a ton of different things that have been proposed for this. I don't have time to go into all of those today. I'm going to tell you about the one I like and then tell you why I think it's important. And so one possible explanation that has gathered some weight behind it recently is that transposable elements actually generate widespread intron gain. So transposable elements are these sort of selfish-genic elements that propagate by making copies of themselves around the genome. And if you're a transposon, maybe right here, where you actually encode your splice sites, right? You've got your DT, your AG, and your otherwise a functional intron. If you're able to splice yourself out of that gene, jump into another exon somewhere else in the genome, you're at a huge advantage because you correctly encode the splice sites. So even though you just inserted in the middle of a gene that might be critical for organismal function, doesn't matter. We splice you right out, the transcript's fine, no one worries about it. So this has been proposed, and in fact, it's actually been observed. And so indirectly observed, of course. And so there's a few wonderful papers on this. I also won't go into all of those. I'm just showing you some nice examples here from Jason Huff's paper in 2016, where what he showed was there were hundreds or even thousands of introns that are almost identical. The sequence logo tells you that these are very, very similar sequences in two different species of algae and a protist. There's also some really nice examples from fungi as well down here, the Vandenberg and Toriani work. And so we had a handful of these. We got maybe 5 or 6 species. And so the question that I started jumping off with, and the kind of question I typically ask is, well, what if I just looked at all the genomes? What would I find? Would I see this as common or not? How would I know? And so the kind of questions we want to answer here are introners the source of many new introns? That is, this is a sort of a dominant mechanism across eukaryotic life. If they are or not, what are the mechanisms that enable their spread? Are they all one kind of transposon, or is there signatures of other things going on here? And what are the processes, sort of broad-scale biological processes that influence their distribution across life? Why should some lineages have thousands of brand new introners and others we don't see any gain between human and mouse over quite a long time? Okay, so taking on the first question first. As many projects in my lab do, we start by developing a bioinformatic pipeline. And the way I do this is I download all the genomes, right? And so at the time, it was a little 8,000-ish genomes. Sanger Tree of Life is cranking these out, so it'd be a ton more today if I was to go back, and I may do that at some point, but let's start there. And if you're interested, I try to put QR codes for these papers on the side here, and they'll be on the next couple slides as well. And this was mostly work, to be upfront about this, mostly work done by a former undergraduate student of mine, Landon Gozashti, who's gone on to have a fabulous graduate career and has kept working on this and collaborating with me since then, which has been just really wonderful to kind of learn and grow with him into this project. And so we start with this. We download all the genomes. We extract all their introns. They're annotated for me, so this is really easy for me to do. And then all I do is an all-by-all comparison. So you can blast them other sequence comparison techniques to basically ask, are there cases where a good portion of introns in this genome look too much like each other, right? And that might be a signature of recent transposition because they have a common ancestor that transposes somewhere else, and I can see that pretty easily. And then, of course, I'll throw some clustering on top of that. Just shown here is one of the ways these things can look when we visualize it. Where basically you're seeing is 2 different introner families that are very differently related to each other, and each node on here would be one introner where the distance is telling me about how similar they are. So there's a whole bunch that are very similar on one side, a whole bunch of greens is very similar elsewhere, okay? And then, of course, lots of QC is thrown on top of this. I don't have time to go through all the things we do to stare at each and every sequence and try to figure out what's really going on here. If you're interested, you can ask me later. I'll say that when you're doing large-scale public data mining projects, the data is messy. It takes a second to clean it up, but we're very confident with the ones we have called here. And then finally, what we're going to do at the end here is once we've got these things called, we have good consensus sequences for each of these. We're going to go back and try to figure out what they are. So we're going to figure out, are they a transposable element? Do they have homology to known TEs? In some cases they do. Are there structural features that indicate something about the mechanism of transposition that we might be able to learn from these sequences? Okay. Okay, so the first thing, just a high level pass, I'll give you kind of a punchline here, is that there's a lot of them. And so what I'm showing you here is a more or less tree of eukaryotic life with bacteria and archaea as my outgroups. And I'm showing the sets, sort of broad scale groups. It's not exactly one phylogenetic level, so bear with me, but broad scale groups where we do find introners are indicated. Where this histogram up top would show you in gray is how many species I looked at. This is log scaled. And then in red is the number of those species within that group where we did find introner elements. And so if we just look at this pretty quickly, there's a lot of stuff over on the left here. And so this would be a bunch of super diverse unicellular organisms and some land plants. So it's things like protists, it's algae, stuff like this. If we keep going, though, we do find that they're incredibly common in ascomycete and basidiomycete fungi. I don't know why, but they are one of the major outliers on this plot. And so there's a ton of them in there. But we do find a few, and people tend to like them. They're definitely unusual, but you find them in animals as well. So we get tunicates and sea urchin as well. So you do find a few in stuff a little bit more closely related to us, but not anything super closely related to vertebrates by any means. Okay, and so from this, we can get a conservative estimate, right? If we just assume these species are randomly sampled, which is not true, then about 2.5% of all species have introners by our definition. I do want to emphasize that that's very conservative, right? We throw out a lot of things that might be introners that we're not sure about. And also, this sampling of species is highly non-random. We don't sample the weird protists in the ocean very much. We do sample the vertebrates that have genomes just like every other vertebrate all the time. And so I think it's very, very likely that this is a dramatic underestimate, but it's hard to know exactly how much. Okay, so from there, we might want to ask something like, what mechanisms are enabling them to spread? And so if we just take a look at this, we say that TEs actually span the full range of molecular diversity. So what I'm showing here is sort of adapted from a review from Cedric Pichot, where they showed kind of the relationships among major classes of transposable elements. And next to that tree, what I'm showing you is both the category of evidence, which I appreciate is certainly not readable, and I'll have to update this slide a little bit, but the various ways that we know what these things are. And then over here is a diagram displaying one of those families that we find. And there's a couple things that you're going to see pretty quickly. Some top one, really nice. We have the polymerase gene. We pretty much know what this thing is doing. It's pretty easy to classify. Others, probably the majority, are non-autonomous transposable elements where you have the terminal inverted repeats, that's those little red arrows up there. But you absolutely do not have any idea exactly what the transposition machinery looks like, even when you search the rest of the genome. And so those of you who have studied transposons before probably will not be surprised by that. Those of you who might be new to this, non-autonomous elements, you often find absolutely tons of them. Autonomous elements, much less. So this is actually as expected, but it's also very frustrating because it does make it a little harder to categorize quite a lot of these. But anyway, you also find, so that's all the DNA transposons, the major classes of DNA transposons. You actually also find basically all the retrotransposons as well, right? So if we go down here, you see retrons, you see non-LTR and LTR elements. Quite a lot of different things pop up. We're able to find almost everything you would expect. But you also get some really nice exotic stuff. So you get some kryptons, and you actually get some helitrons as well. They're certainly rarer, but they do pop up in our data. And so the take-home message from this is that almost any major kind of transposon is capable of generating introns in a genome, as far as we can tell. That doesn't mean every single one will do it, but it does seem like the most of all classes can do it. And if we kind of condense all the data we had and just show you the relative abundances of each of these family types, what you'll see is that something like 80% are DNA transposons with much smaller representation of LTRs and other things. And of course, this is just the ones we can categorize cleanly. That's only about 30% of all introners. And so 70% are something, and it's a little harder to say. I think it probably would reflect the same distribution, and we're just not clear on it, but it's hard to know in advance. Okay. And just to show you some of the most exotic stuff. So even in this single gene here, each of these is a different family of intron, family 3, 2, 9, 74, 9. All of them were found in a single gene in this one species. Now, it's a huge outlier. I pick it because it's interesting, but I do think it helps emphasize just the sheer range of diversity that we're seeing in these things when we look across organisms that have a ton of introner activity in them. How am I doing for time, by the way? Is that clock? Help me.
That clock is a few minutes fast.
Okay. So thank you, just so I have an approximate idea. All right, cool. And so those of you who studied TEs and those of you who haven't maybe wondering, well, how does it always get the right splice sites, right? So TEs aren't such clean copy paste things. They leave lesions in the genome when they insert in many cases. So how are these always splicing correctly? And we spent some time on that too. And so there's a couple different things that go on. In one case, we get things like this, where it really is just a cut and paste. It's got the GT, it's got the AG, it just drops in. It's the exact same sequence every time. And so that one doesn't leave us with a lot of mystery, but the ones you might wonder about, most DNA transposons have what's called target site duplication. What that basically means is you cut the DNA in a way that has sticky ends, right? So a chunk goes here, a chunk goes here. You duplicate that sequence, the transposon goes in between, and because polymerases want to fix this, you fill in the edges, right? So you duplicate the edge and you have a transposon. But of course, if I did that, how would I ever have the AG and GT? And also, I would have an out-of-frame protein potentially. Like something's wrong here, this won't work. And so the way we see this solved in the vast majority of cases is that they actually co-opt one splice site or the other. So essentially, they have to hit a place and create a target site duplication where there's a pre-existing AG or GT. And then that duplication becomes a part of that intron itself. And so by doing this, you have the exact same protein on either edge. You simply got an introner plus a little duplication that contains one of your splice sites in there. And so this works quite well. In fact, the most abundant families we find are doing something like this, which does imply that they miss sometimes and create non-functional transcripts at some rate. Something we're very interested in looking into, but don't know much about yet. There are even lazier transposons, so some that co-opt both. They just have to hit something that goes AGGT. And by doing that, you'll have this target site duplication, but you'll also have both of your splice sites. And then finally, kind of the most exotic and interesting version of this, we get some that actually create new amino acid sequences as well, right? And so what they're doing is they have one of their splice sites, but they also create this large target site duplication and insert a whole bunch of sequence. And as long as it's a multiple of 3, your protein is still in frame. So very often you just get this little local duplication in your amino acid sequence. It may not be, it may not have no functional effect, but there are lots of parts of different proteins that you can hit, add a little bit of sequence in frame, and it probably will not affect very much of the function of that protein. And so this is just one example that was really nice because we were able to go find the orthologous gene from a close relative of this species and show that if you go to that protein sequence, those amino acids are not there. So we're very sure that the introner, or seems very likely that the introner during insertion also inserted those amino acid sequences as well. Okay. Cool. Okay, and finally, going to, I think one of the most interesting questions about this section is, what causes one genome to have introns and one not to have introns? And I think it's really fascinating. As you may recall from that phylogenetic tree at the beginning here, it's really spotty across life, right? There's some groups with an absolute ton of introners, some with much fewer. It cannot be that this is a purely random process. If it were, those numbers would be a lot more even across this thing. There must be some features of some species that promote the gain of new introns in some case. Okay. Okay, and so another way to ask that question would be to ask something like this, which is, what's special about these particular species? If I go to certain groups where they're massively overrepresented, why are those groups different? Okay. And so when we start taking this apart a little bit, what we find is that one of the major correlates, and I think there probably are others, it's hard to know exactly what to look at. That's interesting, but one of the things that jumped out at us very quickly is that there's a lot more introners in marine and aquatic species than there are in terrestrial species. It's a dramatic difference. 17% of the marine organisms we look at, but only 2.5% of terrestrial organisms had introners. And so, and this will stand up to a phylogenetic correction and multiple testing things, all sorts of different ways of looking at this. We're pretty darn sure this is a real signal in the data. The question then is why? And so one thing that we proposed in the first paper and have been able to verify later was it may be that there's horizontal gene transfer that's going on that's facilitating this. So organisms in aquatic environments will tend to be exposed to DNA at a higher rate. So for example, if I get oligos, I put them in salty water to maintain them, right? So it tends to be that DNA is much more stable in aquatic environments. Also, you're much more likely to encounter other cells that are sort of bumping into each other at a fairly high rate. And so this isn't an idea not entirely without precedent. I'd like to give credit to these folks as well. So this is work from Clement Goubert and other folks. But basically what they showed is that if we look across all vertebrate lineages shown here, the fishes in yellow have tons of transfer, horizontal transfer of transposable elements. You can detect it all over this. The vertebrate lineages, which is sort of the tealish color on the edge there, you see very, very little horizontal transfer. And so it's the same kind of concept. They came to a very similar conclusion that probably there's something about aquatic environments that's promoting horizontal transfer, and the rate that you're introducing new introner or potential introner lineages is related to that. But at the time we said this, it was speculation. We had to be very clear about that, and my reviewers were very angry with me about how I phrased this, so we walked it back a bunch. But we, of course, have gone back since then and looked at tons of more examples, and I'll just show you what those look like, where we're able to show, I think, pretty darn conclusively that this is something that's going on here. And so to walk you through this, this is the phylogenetic tree. It's a species tree of ascomycete fungi. And so you'll see the root of it is ancient, right? It's like 650 million years ago, really long time ago. You should have basically no sequence similarity in an intron sequence, which evolved super quickly in that time frame. But if we take all of our introners and we compare them all to each other, we find some really nice things that don't look right. And so this is one of these, where this red lineage, Xylaria, these other less of you, I can't say that, the orange one, they share a common ancestor over about 350 million years ago, that red dot. But when we align those introner sequences to each other, they're about 86% identical between the two groups. So they are extremely similar over about 150 base pairs, something like that. We're pretty darn sure. They're also creating introns in different genes in both species. So they're being introners as far as we can tell in both of these cases, implying that there actually has been a horizontal transmission of an intron creating element. And the reason I show you the fungi and why I really like showing this one is that we actually found another on the same tree, basically, where we have two other species of fungi. In this case, a little more closely related, but still, 115, 120 million years ago, where we see the same thing. There is an introner family that is extremely similar, that has recently horizontally transferred between these lineages and is propagating as new introns in each of these. I won't show it to you today because it will be incredibly boring for you, but we have spent the time to go back and download all the long-read data and really prove to ourselves that these things are really in these genomes. These genes really have the correct species tree. That is, the genes they're inserted into have the same evolutionary relationships as those species do, but those introners clearly do not. They clearly are coming from elsewhere. So we're very, very confident about this. If you'd like to see those figures, you can look at the supplement of this paper and we'll show you, but take my word for it for now. Okay, so that was a really fun one, and it gave us a lot of confidence. And so we, of course, expanded our search, and we found one that I think is kind of even better in some ways. And so in this case, we're looking at what we think is horizontal transfer between a dinoflagellate lineage and a sponge lineage. And so when I show you their common ancestor back here, that is the last eukaryotic common ancestor. It's all the way back in eukaryotes. And yet we have this thing that is actually like 92% identical at the sequence level. And it's really nice because it has propagated a ton in both genomes, right? We've collapsed a lot of elements down here. There's hundreds of elements in this genome and tens in this genome. So again, very, very confident the thing is real and propagating in these genomes. And again, consistent with the idea that recent horizontal gene transfer is responsible for the introduction of an introner lineage into at least one of these two species. Obviously, it came from one to the other or some intermediate. Anyway, something we're really happy about. Okay, so to conclude this section before I go on to some other work here, introners are a really abundant source of introns across eukaryotes. And so again, I want to be clear that every estimate we're giving you is pretty conservative. I do think there's probably a lot more of these things out there that are a little bit less clear in how they work, or they're a little bit more ancient in time and mutation has sort of eroded that signature of similarity that would let me find them. But recently, there have been lots and lots, hundreds, thousands of new introns in various different lineages. Okay. Introns can be created by functionally diverse TEs, and that's in some ways the most important observation of this whole work is that if almost any major kind of TE can create introns, and every eukaryote has splicing machinery, basically any eukaryote could gain some new introns. Now, as I've already shown you, there's huge diversity across life, so that's an approximation, and some lineages do and some don't. But it does mean that introns are sort of almost inevitable, if you will, because you have this splicing machinery that protects your transcripts in some cases when new transposons insert into them. And then finally, one thing we really like, we think that horizontal gene transfer is contributing to this variation in abundance across lineages in various different introner creating strains. Okay. All right. And with that, I'm going to move into the bait and switch part of this, which is also about the origins of genome architecture, but taking an experimental genome structure approach to try to get at this question a little bit. And it's something that we really loved at the time, and his work I kind of shut down in the pandemic, but I'm hoping to fire up again and start working on some more here. So with that, chromosomal inversions, I know it's a bit of a right turn, but go with me. Inversions are structural variants, like they're also genome structure variants. In this case, it's where the map order is reversed, right? So if you're looking at the chromosome on the left here, it goes A, B, C, D in marker order from the acrocentric centromere on down to the telomere. On the other one, it's reversed in the CB region, so it's ACBD, right? So that red chunk is an inversion relative to the other. And one thing that all of us agree about, that is very clearly true of inversions, is that they suppress recombination. And so the reason that works is that if you have a heterokaryotypic individual, that is to say an individual that is heterozygous for an inversion locus, and you form a crossover in the inverted region, then the products just don't work, right? So in the one, you get a chromosome with no centromeres, that's DCBD. The other has 2 centromeres. This is not going to be a happy fit individual in most cases. It isn't quite this simple, but that's not the main point of this talk. So just take my word for it that they do suppress recombination fairly dramatically over large genomic regions. This has led many, many people, including Dobzhansky initially and many others, to come to the idea that inversions are maintaining supergenes. And the idea here is that if we imagine that alleles little c and little b are different from big C, big B, and they have some effect in combination, or they're favorable in similar environments, or they have some relationship to each other that makes them make a better organism, then it can be very favorable to suppress recombination and keep them together. If they were on a freely recombining chromosome, C and B would break up, and you might lose that relationship. Okay. There are tons of examples. I use this one because butterfly wings are involved, but basically you can see this effect in a few different cases. So this is a pretty famous example now from Heliconius literature, where along the top, what I'm showing you is 7 different wings, 7 different species, all from one family of butterflies, Melaniae. And along the bottom are 7 different wings from what's called color morphs, or a long time ago, color races of Heliconius numata, where you can see these are pretty obviously mimicking the ones above them. They all sort of line up 1 to 1 to 1. And this is an example of Müllerian mimicry. Okay, but those are super complex patterns. How are they maintained in nature? And the suggestion is that the reason that these patterns hold true, that you get single morphs that aren't some sort of combination amongst the different morphs within the same species, even though they do hybridize, is that chromosomal inversions maintain the relationships among the mutations that dictate this color pattern. And that's actually been shown. It's a really beautiful paper from a little while ago now, but I just love citing it because it's very pretty. Okay, and that might lead you to some more questions that I'm going to try to answer here. So what is happening at the molecular level, right? So what are the genes involved? Which ones are selected? How can we know? And secondarily, and a little bit more practically, can we use differential expression analysis to identify these things? So it seems like a straightforward way to do it. I can't do QTL crosses because, as I told you, they suppress recombination. So I'm going to need some other approach to do this sort of thing. Okay. And so there's 2 major ways that inversions could impact gene expression. The first is genome structure itself. So for example, if we had a rearrangement that mixed these sort of large-scale topologically associated domains or regulatory regions, we could imagine that you've got sort of a mixture of regulation that isn't quite working, and you get kind of weird gene expression around the edges of the inversion. Secondarily, and a little bit more directly, you could just have direct breakpoint effects where cis-regulatory elements are moved nearer or further from the genes they regulate and therefore cause differential expression, or in some cases, maybe even break genes like this red one here. And the second one, sort of the supergene hypothesis, is that there are mutations in the inversions themselves that are selected to be in combination, right? And that maintains this linkage. But there's a problem. And the big problem, and as I alluded to earlier in the mention about QTL work, is that natural inversions make it such that genome structure and linked alleles are conflated, right? So if an inversion is associated with a trait, you don't know a priori that it's the genes in it. It could very well be the genome structure itself that drives that. How plausible that is up to you, but it is a possibility. And so when a new natural inversion occurs, it not only may grab those important alleles, but it'll actually grab hundreds or even thousands of other mutations that were present along that chromosome that may not be common in that population. And so you'll have this immediate strong association between those mutations and that haplotype. Okay, so that is what led us to this idea. Well, Melanogaster is a model organism. It has some really nice things you can do with it. One of the nice things you can do with it is genome rearrangement by design. And the way this basically works, and I'll be clear up front here as well, this is basically all work of Iskander Saeed, who went on to a PhD at, I think, Cornell with Andy Clark and is now out in the world somewhere else. But the way this worked was we could generate a fly that had what's called flipase recognition targets, or FRT sequences. And what this is are sequences that draw in a site-specific recombinase, that's FLP there, flipase, something, flipase, and it recognizes these sequences, induces recombination between them. And so the way that works is, through genetic crosses, I create the stalk on the top that has these 2 flipase recognition targets. I express flip recombinase, it creates this event, and what comes back out is a chromosome that's genetically identical to the first one. It has all the exact same alleles, but I've now reversed the map order in between. It's B and A are switched, right? Okay, so when I do this, I have the perfect comparison in some ways, right? And so what we did was we compare our natural inversions, which have this combination of structural effects and the linked alleles along them, with synthetic inversions that should be only the structural effects and nothing else, hopefully. Okay, and then of course, we'll throw some RNA-seq at this and do a differential expression analysis on top of that. And again, I won't go into the methods there. You can ask me later if you're interested to know how that works. At the end, what I've done, what we did was do this for 2 inversions, right? On the one is we create a synthetic mimic of the natural inversion IN3RK that I'll call IN3RSK. On the bottom, the same thing with natural inversion IN2LT. IN2LST is the synthetic mimic that it has, that is to say, it has almost the exact same breakpoints. They're not identical to the sequence level because I get my FRT sites in there and all sorts of other stuff, but they're very similar. They're in the same topologically associated domains, and they have, to all reasonable extents, a pretty good structural mimic here. And again, the QR code will take you to the paper if you're interested in this. Okay, so if we do this, and we just initially take a look at this and plot differential expression along the genome, we can answer the first question very quickly, I think, right? So each dot on there corresponds to a gene that's differentially expressed. We're along here, the 5 major chromosome arms of IN3RSK, the synthetic mimic of natural inversion K. We found literally one gene that was differentially expressed. Whereas in contrast, of course, if you go to IN3RK, the natural inversion, we see there are quite a lot of genes that appear to be differentially expressed between the arrangements there. The same thing is true of IN2LST. There's maybe 8 total genes, something like that, that appear to be differentially expressed. And again, hundreds associated with IN2LT. So that kind of answers our first question. Also, just to orient you, I'm plotting the breakpoint positions here with these vertical lines there and there. So this is 3R, where IN3RK is, 2L, where IN2LT is. Okay. So I just did this. There's very little effect. Okay, so we see this pretty large effect where natural inversions affect hundreds of genes. Okay, so we might want to ask a little bit more specific questions. In neither the natural nor the synthetic inversions do we see a case where there's an enrichment for differential expression in the immediate breakpoint surrounding regions of the inversion genome sequence. So it seems very unlikely to us that there's these sort of proximate effects of either topological domains or cis-regulatory elements that we move around. And so that doesn't seem to be the answer. It doesn't seem like genome structure is causing much of this effect. But we do see something really important, which is that there is a big enrichment for differentially expressed genes on the same chromosome arm as those natural inversions occur, right? And so that's something you might expect if an inversion has gathered at least some amount of cis-regulatory elements, either during formation or after formation as a part of continued evolution in the population. And this is fairly significant enrichment. But you see there's also stuff across the rest of the genome, right? And I've gone out of my way to make these stocks as variable and genetically diverse as possible and have very little background that you should share on the other chromosomes. And so what you can learn from that, if you do some back of the envelope math of this, is that something like 80% of differential expression is actually due to trans-regulatory effects rather than those cis-regulatory elements that we think are here as well. And so this isn't that shocking. The number you get with chromosome substitutions in Drosophila is around 2/3 are trans effects. So it's in the range of numbers we might reasonably expect. Okay, and so finally, the other question you might want to ask is, well, is this because of natural selection, right? And so to do that, or to try to get at that a little bit, we're going to use a little bit of population genetic theory. And the big idea here is that over time, sure, most recombinations suppress, but actually double recombination events get into inversions, and they actually over time will homogenize the arrangements. And if you look at FST in the middle of inversions, you'll see that it's actually quite low. That being said, the breakpoints where recombination is completely suppressed or almost completely suppressed, genetic differentiation tends to stay very high, and that's because you can't get even those double recombinations in there. Those things are just different. Okay, so in a neutral model, eventually over time, what you get is something like this, where there's almost no differentiation in the center, and the edges around the breakpoints are highly differentiated between inverted and standard arrangements. Okay, but if we have an allele that's selected, and it's selected to maintain its association with one arrangement or the other, every time one of those recombination events comes in and replaces that allele, slowly natural selection gets rid of it. And so what it does is it looks a little bit like a break, where you'll get an excess of differentiation at that site and around that site, and that will decay rapidly going to the edges away from there. Okay. And so if we look at differentiation along the genome now, where I'm now plotting the same two sort of natural inversions, chromosome arms as dashed lines, and now genetic differentiation along the genome, what you can kind of see if you squint right, and I'll show you some statistics in a second, is that these red points, the differentially expressed points that are on the same chromosome arm, tend to be in highly genetically differentiated regions of the genome here. And so that would be consistent with the idea that that natural selection is maintaining these relationships. In contrast, along the rest of the genome, there's absolutely no enrichment for those other differentially expressed genes, those yellow points, and genetic differentiation, which is what we would expect because, of course, I designed this cross to have no relationship with the rest of the genome. So that would hopefully be true and is. And so again, consistent with the idea that some of this is due to cis-regulatory elements that selection is maintaining, and probably much of the rest of this is due to trans-regulatory effects that are indirectly selected as a part of the inversion. Okay, and so to conclude this section here, so for these inversions, genome structure is probably not the major contributor to expression variation. I think that part is probably pretty darn clear from the work shown. The mutations linked to inversions cause differential expression at hundreds of genes in both cis and in trans. And so we do see a fairly large regulatory effect. I don't think it's a crazy amount. You're talking about 1% of genes or so, but it's substantial across a genome. Okay. And then finally, this correlation of differential expression and genetic differentiation suggests that natural selection is a part of this process that's maintaining these sort of super genes where there's sets of alleles associated with the inversion. And of course, I'm only looking at expression. I think it's entirely possible that other things, protein coding changes, non-coding changes, all sorts of other things might also contribute and be selected. But that's not something we're going to look at with this kind of experimental framework. And so a little bit more broadly, to just kind of try to sum these two things up simultaneously, easy, in quotes, genome sequencing, very possible genome sequencing in functional assays. You make it so that one of the major barriers to getting this information is gone. And actually, it's time for us to go back and ask these questions about why genome architecture evolved in the first place, things that we asked a long time ago and in many cases didn't really answer. And this is, I think I mentioned this last night at dinner. I don't think Anne's here right now. And I attributed it to Dick Lewontin, but Anne told me everyone says this. But the quote I learned from Dick Lewontin was, we don't really answer questions in evolution. We redefine them and we move on to something more tractable. And so I'd like us to take a look back and try to do something a little harder and go back and take on those old questions and try to get there. Okay. And with that, I've got some acknowledgements to do here. So as I said, the two people that really did all the work in this are Iskander and Landon, who both gave me photos of them near water for some reason. And then all of the collaborators and friends that have contributed to these two projects. On the far side, there is my lab the day they all dressed in Hawaiian shirts. And then on the bottom, some funding. And so thank you all.
It's a great talk. I really enjoyed it a lot. I have a bunch of questions, but I'll ask some of them.
Okay.
First of all, do you find, given that your argument about aquatic habitats facilitating horizontal gene transfer, did you see a pattern of greater numbers of introns in the marine organisms relative to terrestrial organisms?
This is an incredibly good question. I'm going to repeat it for Zoom real quick, which is basically, I'm paraphrasing it for Zoom, I guess, that we see this thing where there seems like there's a lot more intron gain going on in these aquatic than terrestrial organisms. Does that process play out such that there's simply more introns in genomes of aquatic organisms over time? And the answer is it's complicated, but it's not obviously true. And so there are the most intron-rich genomes that we know of to date are like Symbiodinium. So that's a symbiont of corals and algae and things like that. And so certainly the outliers are consistent, but I don't think the general pattern is there as far as we can tell. Yeah.
Effective population size, because that would be a competing mechanism according to Michael Lynch.
Yeah. So can we account for effective population size? For what it's worth, I try to do this well. So I don't fundamentally disagree with Mike Lynch that we should have a neutral null model when we start to think about these things. I think that's sort of his main point. The second part he's making there is that, okay, it's effective population size. So he's ascribing a specific mechanism. And I think the main point of disagreement we basically have here is that if this is dominated or at least determined in large part by differences in the rate of gain of new intron creating lineages, then that might be the main signal. It is incredibly hard to do the analysis you're talking about. So the answer is it's really, really hard to simultaneously have a phylogenetic correction and tease out effective population size versus the rate of gain of new introners. And so I would be interested in it, but I think it's probably going to be pretty tricky to do it right for tons of reasons. John?
Do you think there's a correlation just with organisms that release their gametes into the environment, like whether it's plant pollen or fungal spores or aquatic organisms that release gametes into the water?
So there is something else we've looked at. So I'll repeat this question as well. So the question is there a relationship to organisms that are either broadcast spawning or maybe whose germline gets into contact with the environment at a high rate? So we have looked at this. There's a couple challenges. One is sometimes the metadata is easy to get. It was easy to know if an organism's aquatic or not, right? Some of that metadata is a little harder for me to get when I just download a genome sequence. And so it was a little challenging to ask. But one smaller study we did on this was to ask, if we take all the organisms that don't seem to have a sequestered germline and compare them to everything else like vertebrates, et cetera, that seem to sequester the germline, yes, you get a pretty big enrichment where things without a sequestered germline do this. That being said, that's also correlated with the environment they live in. They're more likely to be in the ocean, and it gets pretty confounded. But for sure, I think there are many other factors that determine the rate of intron gain. We put that one forth because It's consistent with the literature. It was relatively easy to show that it is true. And so it seemed like a good starting point, but we have lots of ongoing work trying to figure that out. So great. Yeah.
So I have two questions, I guess, about the intron part. So I guess I'm wondering if the fundamental mechanism could be inherent to the reaction of intron excision or insertion. And so in modern day group 2 introns or the spliceosome, whether or not you excise an intron or insert it back in, is exquisitely sensitive to the salt, the concentrations, and what particular ions are present.
Mhm.
And we can imagine marine organisms, this could be very different than terrestrial organisms.
Oof, okay.
Thought about that.
Yeah, so I'll try to rephrase this question. I apologize if I butcher this, but for Zoom here. So the question is basically, environmental conditions can dramatically affect the efficiency of splicing. So salt concentrations, all sorts of other things that you might be associated with while you're doing these splicing operations. We have thought about this, but that's even harder to get the metadata and to interpret that. And so I think it's possible. It's one of the challenges of when you do this kind of work where you explore as much of life as possible, is that there are a billion things that are correlated with the edges of life that you're finding. And so would love to talk to you more about it. I have not even tried to take on that analysis. The others I'm trying to take on. Yeah.
So pRNAs are responsible for suppressing transposable elements in animals, particularly in germ cells. I'm wondering if there's a correlation between evolution of pRNAs and the associated machineries with introners and the intron propagation.
Right. So the question is, do we see a correlation between these regulatory mechanisms or transposon regulatory mechanisms like pRNAs and the evolution of introners? And the answer is I've never looked, and I should. So I will think about that. Yeah. That's good. Who's got hands up? Yeah. Sure.
Your result intriguing.
Yep.
And my postdoc Zimyo over there has a paper in Nature Communications
Yep.
where we found that around the chromosomal fusion sites,
Mhm.
there was an elevation of ion transporters, and they show signatures of selection. I'm wondering what kinds of genes are around your inversion sites, and do they show signatures of selection? Do you have any population genomic data showing that?
So I have done lots and lots of this kind of analysis in my graduate career. And so, sorry, to repeat the question again, the question is basically, is there signatures, either population genetic signatures of selection in the genes around inversion breakpoints, and do we see sort of differential genes that are there? Like if I was to do geo analysis, I see this. So I've done that analysis. The answer is there is not geo enrichment, or at least not convincing geo enrichment in these regions. The second question of molecular selection, yes, there are in some cases. So I did this in grad school. I don't think I ever published this, but I can tell you that a couple of these are very near to rapidly evolving genes. And so you do see this at some rate. I wouldn't say it was the most common. I think most of them, there was no obvious signature there, but there's a particular inversion I love on the X chromosome that has some stuff around the breakpoint for sure. And so I think it's certainly possible that this is a part of what's going on there. Yeah. Yeah.
Cool.
Good.
Thanks.
Cool, sure. All right. First one in the back.
Yeah, so I wanted to ask, have you found, or do you think is there any group of genes with particular functions that are associated with this introner invasion events of sorts?
Okay, this is a great question. So the question is basically, is there something special about the genes that gain introners, right? Are they different in some way? The answer is we're working on it. That is a very much ongoing chapter of a thesis that I don't think we have a complete conclusion to, but I can tell you that in some of these lineages, yes. One of the major things we see is that genes with relatively low expression levels are way more likely to accumulate introners than those with high expression levels, which I don't think is particularly shocking if they are weakly deleterious at some rates, but there's other things as well. So there are some interesting geo enrichments and things like this that we think are probably important as well. Yeah, cool. That's a good question though. If you ever want to do a rotation in a lab, you should let me know. Cool. Folks.
Let's
Yeah.